The overall goal of this imaging and analysis technique is to isolate specific information from a complex signal. Specifically, this protocol describes how to visualize and quantify nerve-specific mitochondria away from other mitochondria within the epidermis.
This method can help answer key questions in the field of neurology, such as how does mitochondria within intraepidermal nerve fibers of healthy individuals compare to nerve-specific mitochondria of patients with neurological complications?
The main advantage of this technique is that we take a complex signal and extract specific information from it. This protocol helps us to isolate a specific signal, such as the signal in these straws from the signals around them.
Prepare for fluorescence immunohistochemistry staining of skin biopsies by first labeling a 96-well plate according to this schematic. Then pipette 150 microliters of stock signal enhancer solution to reduce nonspecific binding of secondary antibodies into each well of the top row. Prepare rinse wells by adding 150 microliters of 1,X-phosphate buffered saline into each well in rows 2 and 3 of the 96-well plate. Then add 150 microliters of 5% BSA blocking solution into the wells of row 4. Dilute the primary antibodies PGP 9.5 and PDH in 1,500 microliters of 1% BSA rinsing solution and add 150 microliters to each well in row 5.